Mycology. So now we will see the important concepts and probability questions that we can expect from mycology for ASRB Net 2023 plant pathology. So starts with the general basics. Hope you guys can easily understand, because those who are all writing this national eligibility test, they all are mostly MSc graduate, so that this basics will be very easy for you. Like you guys know very well about mycelium, like it's a group of hyphae collectively called as mycelium, and the hyphae, it is a microscopic structure, a thin thread-like structure. So here hyphae is divided into two kinds, that is septate hyphae and aseptate hyphae. Septate hyphae means if the septa is present or if the cross walls is present, it is called septate hyphae. If there is no cross walls, it is called aseptate hyphae or cenocytic hyphae, otherwise called as non-septate hyphae. I have seen this question, like hypha with no cross walls. Which of the following option is suitable for hypha with no cross walls? Non-septate hyphae, cenocytic hyphae, aseptate hyphae, all the above, something like that they will ask. So if the septa having no cross walls, it is called as aseptate hyphae, otherwise called as cenocytic hyphae or coinocytic hyphae you can call, otherwise called as non-septate hyphae. Rhizoids mean anchoring organ of rhizophus. So hope we will discuss this in detail about this. Imagine it is a rhizophus. So in rhizophus, the anchoring organ, this anchoring organ is called rhizophus. This anchoring organ is called rhizophus. So the next one is sporangiospore, sporangiopore. So this stalk-like structure, is called sporangiopore. Imagine, let me draw a diagram. So imagine, hope you guys know that it is a stalk-like structure is called, this stalk-like structure is called sporangiopore. Okay. Inside there is a globo structure is called, right, this globo structure is called sporangia. Inside sporangia, small spores will be there. These spores are called sporangiospores. This is called sporangia. Inside sporangia, sporangiospores will be there. So the next one is homothallic and heterothallic. So homothallic means male and female reproductive organs are present in the same thallus. Example, pythium. So when it comes to heterothallic, male and female organs are not present in the same thallus but on a different thallus. Example is Phytophthora. Hope you guys easily understand. Let's go into in detail topics very quickly. So the next one is Appressorium. We know very well what is Appressorium. That is, Appressorium means attachment organ of fungi. Haustoria means nutrient absorbing organ of fungi. Let me tell you something. Imagine it is a leaf surface. So if the initially spore coming in contact with the leaf, imagine it is a spore coming in contact with the leaf. So it produces a germination tube during unfavorable conditions with a little amount of moisture that is required for the spore germination. So if the spore gets the required amount of moisture under favorable conditions, it produces a germination tube. Okay, after germination tube, it will produce some some globose or some globose like structure. So this is called Appressorium. Appressorium means attachment organ of fungi. So why Appressorium need to form? Because it need to anchor or it need to attach, right? So it need to attach so that it can give very strong connection, where from Appressorium a small hyphae, or what can I say, that a small tube like structure that can enter into the inside the cells. Imagine it, these are all the epidermal cells, circuit, all those things. So inside the cells, this Appressorium will, a small tube will develop from that. This is called penetration peg. From penetration peg, small globose like structures can be produced and enters into the inside cell, that is called haustoria. Here we can see clearly this structure is called Appressorium. Appressorium is attachment organ of fungi, which mainly helpful in the attachment, or which anchors or which strongly holds the leaf surface, from where inside we can see this globose structure, right? This is called haustoria. The function of Appressorium is attachment organ, whereas when it comes to haustoria, haustoria is just nutrient absorbing organ, because the fungi need to develop, right? So not only fungi, any organism, if it need to development or reproduce or it want to develop further, it need food. So where the haustoria draws the nutrients, or haustoria can enters into the inside the cell and it absorb all the nutrients that whatever required for the fungi. Here it is very important, because they will ask questions like, what is the attachment organ of fungi? That is Appressorium. And what is the nutrient absorbing organ or energy absorbing organ of fungi? Something like that there might be a chance of asking. That is haustoria. Here there are different kinds of haustoria. We can see here a shape of haustoria and the pathogen that can produce. Okay, Here Phytophthora starts with Noblic. So Noblic haustoria is produced by albugo. So Noblic means the this kind of structures we can see. Noblic haustoria is produced by albugo. Finger-like haustoria is produced by Phytophthora. See, finger like this. So finger like haustoria is produced by Phytophthora. Branched or pig like haustoria is produced by Sclerospora. Bulb like haustoria is produced by Cuscuta. Wedge shaped haustoria is produced by Archithobium. Whereas finger shaped haustoria is produced by Downy mildew. Powdery mildew produces globose shaped haustoria, and pyriform haustoria is produced by Vegetative. We may expect one or two, like what is albugo produce, which kind of which shape of haustoria, or knob shape, or Phytophthora produce finger shaped haustoria, something like that. There might be a chance of asking this, the shapes of haustoria and their pathogens. So the next one, and this slide is very important to understand the general basics about the spore morphologies, and what are all the different kinds of phylum that can produce spores and all those things. So here motile asexual spore, I repeat, here in fungi can reproduce by means of two: sexual reproduction and asexual reproduction. Sexual reproduction and asexual reproduction. So motile means uh, for our understanding, I can say it can move from one area to another area. It can move from one place to another place. So motile asexual spores of Mycota. So Mycota produce zoospores as motile spores. Zoospores contains flagella. This flagella helpful for the motility or locomotion. Okay, motile asexual spores of Mycota is zoospores, or zoospores otherwise called as motile spores, or planospores, or swam spores. Swam means swimming. So for swimming we are using hands, right? So in the meantime spore uses this flagella. Okay, spore uses this flagella. So motile asexual spores of the fungus is normally known as planospores. Motile asexual spore is normally known as planospores. The zoospores are, we can also call this zoospores as a planospores, because it can use it for motility. And non-motile asexual spores of the fungus is known as aplanospores or aplanospores. Aplano means it can't motile. No, it having no hair, so that it can't move, or no flagella, so it can't move. So the chromista is otherwise called as straminophiles. So the next one is process of formation of spores is called sporogenesis or sporulation. The process of formation of spores is called sporogenesis or sporulation. Okay, so budding spores are called blastospores or blastoconidia. Here I would like to tell some other things. So for example, we mainly divided fungi into so many phylum, right? So let's discuss about some important phyla. So for example, starts with the Mycota. Mycota produce zoospores as asexual spore and oospore as a sexual spore. If it comes to Zygomycota, it produce conidia and as well as zoospore. It produces conidia and as well as zoospore as asexual spore, zygospore as sexual spore. I repeat, Mycota produce zoospores as asexual spores, oospore as a sexual spore. Zygomycota produce conidia or zoospore as asexual spore, and zygospore as sexual spore. And here Mycota and Zygomycota are diploid. Diploid means 2n conditions. So Mycota and Zygomycota are diploid resting sexual spores. Diploid resting sexual spores. When it comes to Ascomycota and Basidiomycota, Ascomycota and Basidiomycota produce asexual spores as conidia, and sexual spores, Ascomycota produce sexual spores as Ascospore, Basidiomycota produce sexual spores as Basidiospores. These ascospores and basidiospores are haploid non-resting spores. Non-resting spores. So in later slides we will discuss in detail about all those things. Let me come once again. Mycota and Zygomycota. Mycota produce zoospores as asexual spores, oospore as a sexual spore. Zygomycota produce conidia and zoospore as a asexual spore, zygospore as a sexual spore. This oospore and zygospores are diploid resting spores, resting sexual spores, whereas ascospores and basidiospores are haploid non-resting sexual spores. So the next one is a various type of septa that is found in the fungi. So we will see the 5 kinds of septa we can see here. So solid septa and perforated septum, or bordered pit type of septum, ascomycetian septum. The last one is Dolipore septum. Now I will tell you just some general facts about this septas. Hope you guys know very well about this, and let me tell you some general facts. In solid type of septa, it is a very simple type of septum, right? So with cross walls, we can see here it is a very simple type of septum with cross walls. Cross walls are present. So here it is produced by the lower fungi like Mycota, such kind of fungi that can produce. So it to delimit the older and reproductive parts of the mycelium. So the mainly it is a simple type of septum, helps in the delimitation of older and reproductive parts of the mycelium. So it is a solid shape, solid type septum, and Mycetes can produce. When it comes to the next one, second one, perforated septum, we can see perforated in the sense simple perforated by number of bodies. We can see simple perforated stuff, we can see here small bits of perforated septum can be seen, and this type of septum can be observed in geotrichum. The next one is bordered pit type of septum. So actually this we can see here a small pore, this pore can be seen, right? So it is actually surrounded by bifurcations or overarching bifurcations can be seen in this border type of septum. So this is helps in like blocking the pore, but the transportation of material from one side to another side, so it can simply block based on the requirement of the mycelium, based on the requirement. So this type of septum can be observed in mucorales and as well as maybe trichomycetes, I think. Make sure of it. And the next one is the Ascomycetian septum. Mostly we can see this type of septum in Ascomycetes as well as Deuteriomycetes. And the last one is a very important one, is Dolipore septum. So they can ask questions like, what is the function of Dolipore septum? So you just to the main function of Dolipore septum is to ensure the dikaryotic condition, because if all the nuclear material or protoplasmic material move from one side to another side, one side become completely empty, so that the mycelium may dry, right? So to just ensure the dikaryotic condition, this Dolipore septum will be helpful. So for if I want to tell you, it is a barrel shaped structure. We can see it is a barrel shaped hemispherical structure. You can also say barrel shaped hemispherical structure. This hemispherical structure is called parenthosome. So okay, this hemispherical structure is called parenthosome. This function of Dolipore septum is to ensure the dikaryotic condition of the mycelium or dikaryotic condition of the hyphae. So that was the pore cap, otherwise called as I told you that parenthosome. So mostly we can see this Dolipore type of septum in Basidiomycota group of fungi to ensure the dikaryotic condition of a mycelium or hyphae. The next general information about the fungi is the nutrition type of fungi. We know very well fungi, that is absorption, and algal cell wall is made up of. In the general plant pathology itself, we already discussed that fungal cell wall is made up of chitin and a little amount of glucan. Algal cell wall is made up of glucan and little amount of cellulose. And cell wall less pathogen, so molecules, that is a phytoplasma and spiroplasma doesn't contains any cell wall. Instead of cell wall, it contains a cell membrane, 3-layered or triploblastic cell membrane can be observed. So in virology we can discuss about all this phytoplasma and spiroplasma. I think in bacteriology also there we'll see. And chitin containing Mycetes fungi, we know very well that Mycetes group of fungi. So if you guys are a little bit confused about this, in general plant pathology, go to this playlist and you just find out these videos. We are publishing one by one, like general plant pathology, mycology, bacteriology, viral, something like that. You go to general plant pathology, you guys go to general plant pathology. We have given the cell wall compositions of fungi, bacteria, Mycetes and various other kinds of important fungi. Normally fungi contains chitin and a little amount of glucan in their cell wall, but chitin, Mycetes generally contains cellulose and a little amount of hydroxyproline can be observed. So little amount of hydroxyproline, but normally Mycetes group of cell wall is composed of cellulose. But few of the fungi that even though that comes under Mycetes, their cell wall is made up of chitin. That's why they are calling as chitin containing Mycetes fungi. I have seen this question many times, so I think it will be very important. So Acaila, Aphadogyla, Saprolignia and Leptometus, these 4 fungi are, even though they comes under Mycetes group of fungi, but their cell wall is made up of chitin. Okay, So the next one is the chitin is a compound of N-acetyl glucosamine units. So reserve food material or storage product of fungi. I think this was the reserve food material of fungi, Mycology, red algae. All those things were already mentioned in general plant pathology in a table form, I think. So let me come once again. The reserve food material of fungi or storage product of fungi is glycogen, or the fungi can store their food in the form of glycogen or in the form of oil or maybe in the form of lipids or in the form of oil globules. So the next one is the reserve food material storage organ of Mycota, that is mycolaminarin. So the next one is the reserve food material storage product of red algae. Red algae, it is a laminarin. The next one is the flagellated protozoans belonging to the genus Phytomonas. So the next one is example of haplodiplobiontic yeast. So example of haplodiplobiontic yeast is Saccharomyces cerevisiae. So when it comes to what is haplodiplobiontic life cycle, both the haploid and diploid phases are equally important in the life cycle, so that we can call it as haplodiplobiontic life cycle. I mean haplodiplobiontic example of haplodiplobiontic yeast is Saccharomyces cerevisiae. It has been, I think it has been asked before, and only one yeast that reproduce by means of fission. So most of the yeast that reproduce by means of budding, but one yeast is called that can reproduce by means of fission, that is also called as fission yeast, that is Saccharomyces also called as fission yeast, that I told you. And the next one is give me an example of fungi which do not produce Appressorium and haustoria. So when it comes to Pythium do not produce any Appressorium and any haustoria. It is like gametangel contact type, right? So most of the yeast are deficient in the vitamin. So all yeasts are deficient in the vitamin, that is H, biotin. Okay. Alternate of haploid and diploid generations are found in Saccharomyces cerevisiae. That is alternation of haploid and diploid generations are, that is haplodiplobiontic life cycle. I think we already seen that is haplodiplobiontic yeast example is Saccharomyces cerevisiae. Alteration of haploid and diploid generations is called haplodiplobiontic life cycle. So the attachment portion of flagella is called, so imagine, so it is imagine if it is a flagella, it is a fungal flagella. So this attachment portion is called as blepharoblast. So rope-like strands of hyphae is known as rhizomorph. If you see the rhizomorph, so the rhizomorph is like a thread-like strands or rope-like strands. That's why it's called as rhizomorph. So which fungi from rhizomorph as a resting spore, act as a resting spore? So Armillaria melia and Ganoderma lucidum. So rhizomorph is otherwise called as mycelial cords. This one is very important. Sclerotium producing. So hope you guys are understanding what I am trying to tell, because since all of the students are MSc at least MSc graduated, so that they have at least minimum basics. So that's why I am going a little bit fast. Hope it will be okay. And Sclerotium producing genera under unfavorable condition. So under unfavorable conditions, some pathogens produce Sclerotia. So this Sclerotium can be used to survive during unfavorable environmental conditions. Whenever the favorable environmental conditions will be back, or whenever the favorable environmental conditions will return, the Sclerotia can germinate and give rise to new mycelium. It is a Sclerotium is a very black color or sometimes brown color, hard resting structure. It's like a mustard shaped, and also look like a mustard also. Okay, and it is a hard black color or brown color, hard resting structure that is produced by some fungi. Now we will see what are the fungi that can produce Sclerotium during unfavorable conditions. So first example is Sclerotium ralphsi, Rhizoctonia, Sclerotinia, Claviceps and Macrophomina. These are all the fungi which produce Sclerotium during unfavorable environmental conditions. I repeat, it's a very important. Sclerotium ralphsi, Rhizoctonia solani, Sclerotinia, Claviceps species or Claviceps species, and Macrophomina fasciolina or Macrophomina. So now we will hope you guys know very well about sexual reproduction and asexual reproduction. I don't want to take much more time about this. So let me tell you in general way. So asexual reproduction is of two types, that is the vegetative types and normally for the production of spores and all those things. So let's go through vegetative type. So the first one is the fragmentation. Very simple. A small fragments of hyphae broke into small bits, and each bit can have a capability so that they can develop individually and give rise to new mycelium or new fungi. So here the hyphae broke into very small pieces. Very small pieces. This species is otherwise called as idea, otherwise called as arthrospores, we can see here, we can see here. So the small spore hyphae break into small spores or small fragments, that's called idea or arthrospores. So like fungi like aspergillus and rhizophus, this type of fragmentation can be observed. The second one is a budding, mostly we can observe seen in yeast. A small fructification, imagine it is a yeast. A small fructification can be developed on some side, and this the nuclear material will be transferred into this small fructifications or bud or outgrowth. So later this small cell will detach from the mother cell and starts to behave like a new budding cells. So mostly we can observe like yeast and bacteria also. Most of the plant pathogenic bacteria also can reproduce by means of budding. Okay, and coming to the fission very simple, where the cell is divided equally and whereby forming a constriction in between. So I told you already, fission yeast, Schizosaccharomyces or fission yeast. And like rhizomorphs also, as I told you, like it's like like a small rope-like structures or thread-like structure that is produced by the few fungi like Armillariella millia and one more Ganodermal eustums. And the last one is the next one is the Sclerotium. Now itself we discussed before, just before in the previous slide only we discussed. It's a hard restring structure which is actually brown or black in color, which contains enormous amount of food material so that it can survive for a very long period in the soil during unfavorable environmental conditions. Whenever the favorable environmental conditions are onset of the favorable environmental conditions, the same Sclerotia will germinate and give rise to new fungi. Now when it comes to the asexual reproduction, like by means of production of asexual spores, we will see. So this is like, first one is the Sporangia or Sporangiospores. See, this is this dome shaped structure or sac like structure is called Sporangia. Okay, so this holding a stalk like structure which is holding is called Sporangiospore. I think we discussed in the first slide itself, right? So inside sporangia, small spores will be there. This small spores is called sporangiospores. This sporangium, inside sporangium, a small spores can be packed inside. This is called sporangiopores. For example, we discussed already aplanospores and planospores. Planospores means motile, aplanospores means non-motile. Zoospores are like planospores that can able to motile, right? So these aplanospores and zoospores are actually developed from this, or can be produced by this sporangiospore type. And when it comes to the next one is this is the zoospores, where zoospores are mostly look like kidney shaped, right? That I will tell you why I just drawed only kidney shaped zoospores here. And the next one is the conidia type. Conidia or conidiopore, that is idea, where the hyphae is break up into a small fragments or small like spores. This is called these individual spores are called idea. Each and every individual spore can give rise to a new fungi. This idea otherwise called as arthrospores. Idea is otherwise called as arthrospores. And the next one is the chlamydospores. So chlamydospores is also asexual spores, so that is produced by some fungi like Fusarium, mucar and various other fungi. So it is also a thick walled resting spore, we can see here. It is also a very thick walled resting spore that can be produced by the fungi like mucar and as well as Fusarium. So when it comes to the sexual reproduction of fungi, so as I told you in previous slides, the fungi can reproduce by means of sexual and asexual reproduction. So now we will see what are the sexual reproduction. When it comes to sexual reproduction, there is a sequence that is plasmogamy, karyogamy, meiosis. So hope you guys know very well, and let me go a little bit fast. Okay, plasmogamy, karyogamy and meiosis, where plasmogamy means sexual reproduction between the union of two protoplasms. Here plasmogamy again divided into many types, that is gametogamy, otherwise called as planogametic copulation. In planogametic copulation we can see this isogamy. Iso means same, gamy means fusion. So the fusion of same kind of gametes, which is morphologically and physiologically similar, that is isogamy. So isogamy can be observed in the fungi like Oilphidium and Syncytium. And anisogamy means not similar. So fusion of
Dissimilar gametes are morphological and physiologically dissimilar gametes, that is called anisogamy. We can observe in allomystis and blastocladians. So when it comes to heterogamy, fusion of motile male cells with non-motile female cells. Okay, here male cells are motile, whereas female cells are non-motile. So we can observe this in monoblepharis. The next one is the gametangial contact type. We are talking about now plasmogamy. Okay, in plasmogamy different types are there, that is gametogamy. The next one is gametangiogamy. It is otherwise called as gametangial contact. When it comes to the gametangial contact, male reproductive organ is called antheridium, whereas female reproductive organ is called oogonium, where male and female reproductive organs are coming in contact with each other, and there will be a small fertilization tubes developed from male to female, where this antheridium, that is the male reproductive organ, passed to female. This is how the gametangial contact works. So just very simple, gametes are coming in contact with each other. Male gamete called antheridium, female gamete is called oogonium. Okay, they are coming in contact with each other, and the genetic material, or the, what can I say, the gametes are exchanging from male to, or gamete is passing from male to female gametes. So this type of gametangial contact can mainly observed in albugo, pythium and phytophthora, like Mycology group of fungi. Okay, so coming to the next one is gametangy, or otherwise called as gametangial copulation. That is gametangy, or otherwise called as gametangial copulation. Now we will see again in gametangial copulation, three types will be there: hologamy, direct fission, and anisogamy. Very simple. The whole idea, I mean the whole ideology behind this is very simple: fusion of entire content of two gametangia. That is, imagine it is a gametangia, right? So the fusion of entire content of two gametangia, where gametes are coming in close with each other, and fusion takes place in a different ways. So generally few two gametes are coming in contact, and the genetic material will be exchanged, like the methods of exchange may be hologamy or direct fusion or anisogamy, maybe. The next one is spermatization. So spermatization means it is a kind of sexual reproduction, and where in the upper side we can see flask shaped structure, that is called spermagonia. Okay, flask shaped structure is called spermagonia, which can be seen on the upper surface, and this can produce both kinds of positive, I mean like plus and minus gametes. I can say it's for our understanding, I can say opposite gametes, like plus and minus gametes. So where insects are involved in transmission, maybe wind and water also involved in the transmission of this, and where male and female plus and minus coming in contact, and where spermatization actually takes place. For example, this kind we can mainly observe in Puccinia graminis tritici. The last one is somatogamy. So the fusion of vegetative cells or somatic cells. Very simple. The fusion of somatic cells or vegetative cells is called somatogamy. So we can observe, especially like some basidiomycota group of fungi, like agaricus, pezziza, something like that. So agaric basidiomycota group of fungi, we can observe this. So this is about sexual and asexual reproduction. So coming to the fungal flagella, so in previous years I have seen some questions, especially from this fungal flagella section. So let's have some general basics about fungal flagella. We know very well. So fungi flagella actually used for locomotion. So let me tell you, here I am drawing two flagellas. So 9+2 arrangement can be observed in fungi, whereas 9+1 arrangement can be observed in bacteria. 9+1 or 2 arrangement means very simple. Here 9 rings will be there. In the middle only two rings will be there. That is 9+2 arrangement of flagella. If we take the cross section of the flagella, 9+2 arrangement. 9 rings at the corner, 2 rings at the middle. So the same arrangement was absent in bacteria. Instead of 9+2, 9+1 arrangement can be seen in bacteria. So let's have a basics. So type of flagella, whiplash, otherwise called as acronematic. Whiplash means flagella without hair or flimmers. You can see here whiplash, right? Whiplash means flagella without hair or flimmers. So tin cell, otherwise called as pantonymatic. Tin cell means flagella with hair. Okay, flagella with hair. So if the flagella is in upward direction, it is called anterior. If the flagella is in downward direction, it is called posterior. I have given here. So anterior. If it is anterior, upside. Downside means posterior. So now we will see. So by having the basics, we will see. So here we can see here. So this is a flagella, right? This is a flagella. Here the flagella having no hair and it is a downward direction. If the flagella is having no hair, it is called as whiplash and it is showing a downward direction, which means downward means posterior, upward means anterior. So now we can say this as posterior whiplash. Posterior means it is showing downside. Whiplash means no hair because there is no hair, right? So posterior whiplash type of flagella can be observed in Chytridiomycetes. Chytridiomycetes, it's a very important. All these flagella types are very important. There might be a chance of asking questions, okay? And we can see the flagella is upward direction. Upward means anterior. And it is producing hairs, right? And it is producing hairs. That's why it's called as tinsel. So we can call it as anterior tinsel because anterior means upside, hairs are there. That's why it's called as tinsel. So anterior tinsel type of flagella is produced by Hypochytridiomycetes. The next one is here two flagella is there. Both flagella is upward direction only. One flagella is short and one flagella is lengthy. And here we can say anterior because the flagella is upward direction. Anterior whiplash type biflagellate. Why we are saying biflagellate? Because two flagella is present. Okay. Biflagellate with anterior whiplash. Whiplash means no hair. So biflagellate with anterior whiplash type of flagella can be observed in Plasmodiophoramycota. Here in whiplash, one is short, one is long. Then last one is here we can see it is a kidney shaped, right? So anterior one flagella is there with hair and posterior another flagella is there without hair. Now we will start with the anterior. So if it is hair, what we are calling it is tinsel. If it is no hair, it is whiplash, right? If now it is anterior tinsel, posterior whiplash. Anterior tinsel and posterior whiplash type of is the characteristic feature of oomycetes. Is very important. I have seen this question in many places, even in exams and many exams. It was very important. They will ask questions like this: what is the special feature of oomycetes flagella? Biflagellate with anterior tinsel and posterior whiplash is the special character. Okay. Anyway, let me tell once again. If the flagella is upside, it is called anterior. If it is downside, it is called posterior. If flagella having no hair, it is called whiplash. If the flagella having hair, it is called tinsel. So in Chytridiomycota produce posterior whiplash type of flagella. When it comes to Hypochytridiomycetes, it produce anterior tinsel. Anterior means upside, tinsel means hair. Plasmodiophoramycota produce biflagellate. One is short and one is long. The two anterior whiplash type. Whiplash means no hair. And oomycetes special character is produce anterior tinsel and posterior whiplash type. So these are all the special characters. These are all the special information related to fungal flagella. As I told you, we already discussed before. Ascospores and basidiospores, whatever the spores that I have given, all are sexual spores only. Okay, but ascospores and basidiospores are haploid, means N condition. Haploid, non-resting spore. They are not a resting spores. It is a non-resting spores. When it comes to oospores and zygospores, they are diploid in condition, 2N. Diploid in condition and they are resting spores. They will ask questions like this: which of the following is a diploid non-resting spore? A. oospore, B. basidiospore, C. ascospore, D. all the above. Something like that they will ask. So ascospores and basidiospores are haploid non-resting spores. It is not a resting spores. When it comes to oospores and zygospores, we know very well oospore and zygospores are resting spores, right? So oospores and zygospores are diploid resting spores. And I have seen the questions in previous exams. I mean net exams only. So let's see the differences between the asexual fruiting body. Asexual fruiting body means very simple, where the asexual spores are actually produced. So first one is the pycnidium. So pycnidium is actually a flask shape. See, it is look like flask shape, right? So it is look like flask shape. So flask shaped structure, the shape of there might be a chance of asking the shapes also. So what is the shape of pycnidium? It is a flask shaped. It is actually a flask shaped structure with a narrow mouth. If you can see, the narrow mouth is called ostiole. We can also expect this kind of question. What is the narrow mouth of a pycnidium? It is called ostiole. Okay, it is also important. I have seen not in exam, net exam, but in somewhere I have seen this question. What is the opening region or opening mouth of pycnidium? That is ostiole, something like that we can expect. So in pycnidium is actually a flask shaped structure. It is a hollow, actually flask shaped globo structure with a circular mouth, right? It is a circular mouth where the spores are produced inside. So examples is also very important. They can also ask examples like this. So pycnidia can be produced by which of the following fungi, or maybe by giving the fungi name, this fungi can produce which kind of asexual spore, something like that they will ask. So the macrophomina is produced, pycnidium is produced by the species of macrophomina, fasciolina and diplodia, and other species also there like Phoma, Ascospore and Septoria. I repeat, pycnidium is produced by macrophomina, diplodia, Phoma, Phoma, Ascospore and Septoria. These are all the fungi that can produce pycnidium. Actually developed from the pycnidium. So the next one is Sporadaceum or Sporadacium. It is actually look like a cushion shape, right? It is actually look like a cushion shaped. So the shape will be the cushion shaped, and it is actually it is looking like a hemispherical or barrel shaped structure with a compound conidiospores, right? From the conidiospores where the conidia is produced. So this kind of asexual fruiting body is produced by Fusarium, Epicoccum, maybe tubercularia, some kind of fungi they can produce. But very important fungi is definitely Fusarium. So Fusarium can actually produce it from the Sporadacium, okay? Sporadacium or Sporadacium, whatever you want to pronunciate. The next one is Asarvulae or Asarvulus, okay? It is actually soccer shape. So it actually normally soccer shape. Soccer shaped depressed pseudoparenchyma cells can be seen. So it can produce it is also like crescent shaped conidia, right? That can be produced by very important one, Colatotrichum and Pestalotia. So Acerulus, so Colatotrichum and Acerulus can be developed from this Acerulus. So the next one is the cinema. We can see here cinema is erect branches. It's looking like a branches, right? So it is a loose aggregation of branches or unbranched erect conidiospores. So all the conidiospores are loosely aggregating and coming together. Loosely aggregating and look like a tree or bunch of trees, like, right? So this kind of cinema is actually some fungi like ceratocystis or maybe graphium or podosporium, some kind of fungi can be produced by this fungi cinema. Let me go fast once again. Pycnidium, it is actually flask shaped structure. Fungi like macrophomina, fasciolina, diplodia, fungi like macrophomina, fasciolina, diplodia, Phoma, Ascospore and Septoria can be produced from this pycnidium. When it comes to Sporadacium, it is actually cushion shaped hemispherical barrel structure, and fungi like Fusarium, Epicoccum and maybe tubercularia that can be produced actually from Sporadacium. Coming to the next one, Acerulus, it is actually soccer shaped depressed pseudoparenchyma aggregation of tissues. As fungi like Colatotrichum and Pestalotia can be produced from this. And the last one is the cinema, or otherwise called as cinemata. Okay, it is like erect branches, conidiophore, aggregation of erect branches of conidiospores. Fungi like ceratocystis, graphium can produce this kind of asexual fruiting bodies. So the coming to the next one, other names of fungi or fungal groups. So this is also very important. They will ask Mycology is otherwise called as, maybe sometimes powder mildew fungi is otherwise called as. Sometimes I have seen the question, that's why I just placed this table. Okay, so other names of fungi or fungi like organisms we will see. So Mycology is called as egg fungi because they produce zygote, that is like a egg like diploid condition. That's why it is like look like a egg. That's why it's called as egg fungi. Pseudo fungi, but it is a lower fungi. It is also called as, we know very well that Mycology fungi is also called as pseudo fungi and also called as zoosporic fungi because in their life cycle zoospores are produced. That's why it's called as zoosporic fungi. And zygomycota is called as yolk fungi because it produces zygote, that is called yolk, otherwise called as zygote fungi. Ascomycete is called as crown fungi or otherwise called as sac fungi. Why it is called as crown fungi? Because when it comes to the entire kingdom or phylum, Ascomycete is the biggest phylum or the largest phylum, which holds around 60 to 70% of the known fungi, right? I mean discovered fungi, that is especially that comes under Ascomycete. That's why it's called as crown, like a king having crown, right? That's why if it is a, I mean king means because he is the man who dominates everything. So that's why Ascomycete is the biggest phylum. That's why it's called as crown fungi. Otherwise called as sac fungi because it produces sacs. Because it produces sacs, right? If you see some fungi, fungi produce sacs. That's why it's called as sac fungi. When it comes to Basidiomycetes, it is called cup fungi or club fungi, where these basidiospores are produced from a the structure is called club. That's why it's called as club fungi, otherwise called as cup fungi. So Deuteromycetes, otherwise called as dustbin fungi. Why it is called as dustbin fungi? Because there is no asexual stage. Sorry, there is no sexual stage. Now this Deuteromycetes is just joined to Ascomycetes. Before it is separated, Deuteromycetes, otherwise called as fungi imperfecti. Fungi imperfecti or dustbin fungi. You can also note it down. Not only dustbin fungi, it is also called as fungi imperfecti. So the next one is the downy mildew fungi. Downy mildew fungi is called as hairy fungi, whereas powdery mildew fungi is called as sac fungi. Actinomycetes, it is called as ray fungi. Pezizales is called as cup fungi because the Pezizal group will produce a cup-like structure. That's why it's called as cup fungi. I repeat once again. Oomycetes is otherwise called as egg fungi or pseudo fungi or zoosporic fungi. Zygomycetes yolk fungi or zygote fungi. Ascomycetes crown fungi or sac fungi. Basidiomycetes cup fungi or club fungi. Deuteromycetes dustbin fungi. Downy mildew hairy. Powdery mildew sac. Actinomycetes ray fungi. Pezizales cup fungi. So yes, asexual stage and sexual stage, as we discussed earlier, that fungi can reproduce by means of sexual reproduction and asexual methods. So when it comes to the asexual stage or asexual fungi is otherwise called as anamorphic fungi, otherwise called as mitotic fungi, because mitotic division will happen, right? Otherwise it produces mitospores. That's why it's called mitotic fungi. Otherwise called as imperfect fungi, because if there is no sexual stage, it is called as imperfect fungi. Otherwise called as residual fungi, otherwise called as secondary fungi. Asexual stage otherwise called as anamorphic stage, mitotic stage, imperfect stage, residual fungi, secondary fungi. These are all the other names of asexual stage. When it comes to sexual stage, it is called as teleomorphic stage, otherwise called as meiotic stage. In sexual reproduction, meiosis takes place, right? That's why it's called as meiotic stage, where meiospores are produced, otherwise called as perfect fungi, otherwise called as perfect fungi. Teleomorph plus anomorph equal to holomorph. Holomorph means teleomorph plus the combination of sexual and asexual cycle. The combination of teleomorph and anomorph is called holomorph, where many asexual cycles or many anomorphic stages can be seen, but only one teleomorphic stage can be seen in holomorph. And the sexual spores are otherwise called as meiospores because they are developed through a process meiospore, right? So examples of sexual spores are oospores, zygospores, ascospores and basidiospores. In the previous slide I told you oospore and zygospore are the sexual resting spores, diploid sexual resting spores, whereas ascospores and basidiospores are the haploid non-resting sexual spores. When it comes to asexual spores, mitospores actually developed through the process of mitosis. Maybe we have already seen zygospores, conidia, oidia, spermatia, micron, macron, all are comes under asexual spores only. So we can expect one question, maybe directly or indirectly we may expect, I'm not sure, we may. And one important thing we need to discuss here, that is some technical terms. Haustoria, otherwise called as sinkers. We know very well what is haustoria. Nutrient absorbing organ of fungi is called haustoria, otherwise called as sinkers. Teleospore that was produced in rust life cycle. Heteroecious rust, autoecious and heteroecious rust. That I will tell you what is autoecious and heteroecious rust in the later slides. Symbiosis or symbionts. Sex hormones, because sex hormones can be produced in fungi also. So fungi also produce some kind of sex hormone to attract gametes. So sex hormones and obligate parasitism and endophytes, all the terms are coined by Anton de Berry, none other than the father of plant pathology. father of plant pathology. Haustoria, teleospores, heterogeneous rust, symbionts, sex hormones, obligate parasitism, endobiotes, all the terms were coined by Anton de Berry. So the next one is the Phytophthora sporangial proliferation. So we will see mainly three types of sporangia can be seen: papillate sporangia, semi-papillate sporangia, and non-papillate. See, papillate means we can clearly see distinct papilla. You see, we can see here. See, we can clearly see some, nodule-like structure. For your understanding, I'm telling it's not a nodule. Just for example, I'm telling, okay, some nodule-like structure or papillae can be seen at the distal end. So that is called papillate. Presence of papilla. Okay, semi-papilla means partial papilla. It's not that much development. See, papillate. So like small papilla, that is not well developed papilla, can be seen. That is semi-papillate. Non-papillate means no papilla. No papillae on the spores, we can see here, and these are the microscopic observations. So papillate means that at the end of the spore there will be a papilla. Examples: Phytophthora nicotiana, Phytophthora capsica, and Phytophthora citropthera can produce this papillate type of sporangia. And coming to semi-papillate, very important one, the Phytophthora infestans. The questions may ask like this: Phytophthora infestans which produce which type of sporangia? A. Papillate, B. Semi-papillate, C. Non-papillate, D. All the above. Something like that they will ask. So Phytophthora infestans produce semi-papillate, whereas partially developed papilla. The last one is the non-papillate. For a Phytophthora cinnamome produce non-papillate type of sporangia. So we will see what are the different kinds of diseases caused by Pythium. So damping up of vegetable caused by Pythium panadermatum, damping up of tobacco and chili caused by Pythium dibarianum, rhizome rot of turmeric caused by Pythium graminicolum, and rhizome rot of ginger which is caused by Pythium myriotalium, and seedling blight of castor, leaf blight of sesame, very important one, seedling blight of castor or castor seedling blight is caused by Phytophthora parasiticae. So this disease was first reported by J.F. Dasher, very famous scientist, and is also the second president of Indian Phytopathological Society, and he is the scientist who extensively worked on Phytophthora in castor, and is also renowned to establish the Phytophthora genus in castor. He's a very famous one. And when it comes to the history section, let me tell you many more interesting and important facts about each and every single scientist. Now we have no time to explain here. And next one is Coiroga of areca nut, which is caused by Phytophthora arecae, and bud rot of palms and citrus gummosis caused by Phytophthora palmivora. See, not only this species, some other species also responsible for the disease, but I just mentioned some important species. Okay. So when it comes to the next one, different types of downy mildew. So actually here the downy mildew is mainly based on the arrangement of conidiospore. Okay, here Sclerospora type. I'm simply telling how the conidial description along with conidiospore description along with the example only. We may expect they will ask questions like that. I will tell you how what will be the probability and how they can ask a question, something like that. So Sclerospora, this is the Sclerospora. If we clearly observe this conidiospore, we can see swollen, long, stout with upright branches. So branches are upright branches. So the fungi like kambu, downy mildew, that is Sclerospora graminicola, can produce this type of sporangiospore. The next one is Peronospora. Peronospora means dichotomously branched at acute angle. You can see here dichotomously branched. All are dichotomously branched at acute angle, right? So such kind of sporangiopore can be observed in onion downy mildew, that is Peronospora destructor, sorghum maize downy mildew, that is Peronosclerospora sorgi. So the next one is Plasmapara. So in Plasmapara, sporangiospores are right angle with blunt tips. You can see here right angle with blunt tips. All the sporangia are right angle, right exactly opposite. That is right angle with the blunt tips. Tips are blunt, right? So grapes downy mildew, that is Plasmapara viticola, produce this kind of sporangiospore. And Basidiopora, club shaped with swollen head. We can see here it is actually club shaped with swollen head. Head is swollen, right? So club shaped with swollen head. Best example is Basidiopora. And the last one is Bremia. So the Bremia, the tips of the branches, you can see here, tips of the branches are actually cup shaped with 4 sterigma. So each branch contains 4 sterigma. Okay, so cup shaped with 4 sterigma. Example, lettuce downy mildew, that is Bremia lettuce. So I will tell you what will be how the questions they can ask. So they will simply ask Downy Sporangiospore are dichotomously branched with acute angle is the special feature of which fungi. So here we can see dichotomously with dichotomously branched with acute angle is the special feature of Peranospora. So if it is a right angle with the blunt tips, it is the special character of Plasmopora. They can also give like this. So Plasmopora viticola produces a sporangiopore which is of right angle with blunt tips, or maybe they can ask questions like this: Plasmopora viticola produce which kind of sporangiopore? Right angle with blunt tips, dichotomously branched, swollen head with stout, or something like that they can ask. So you please go through this, it is very very important. I have so since years I have lots of confusion on this, especially this downy mildew topic only. Now I was much better and clear about this downy mildew and all the arrangements. You guys just please go through. So the next one is disease caused by Phytophthora, and I have mentioned so many, so I don't think that it's not necessary to just study all those things. That is in rubber abnormal leaf called, that is called Phytophthora palmivora, and red gum stem blight, castor seedling blight, tobacco black shank, which is called by Phytophthora parasitica. These are all the different kinds of diseases in various crops that is caused by Phytophthora species. So you just I think it's not necessary to study because time is going up. So you guys hope it will be very easy to, if you guys can understand if you go through. So the next one is different types of sexual fruiting bodies. So sexual fruiting bodies where sexual spores are produced. I will tell you in a very simple way. Completely closed ascocarp, we can see here completely closed ascocarp is called Cleistothecium. So Cleistothecium means completely closed ascocarp is called Cleistothecium. So when it comes to completely opened ascocarp, it is called Apothecium. So completely opened ascocarp is called Apothecium, whereas partially opened ascocarp is called Perithecium. We can see completely opened ascocarp is completely opened here, whereas partially opened ascocarp is called Perithecium. Partially opened ascocarp is called Perithecium, whereas not fully or partially closed ascocarp is called Gymnothecium, or we can also naked ascus type, naked ascus type. So let me tell you, if it is a completely closed ascocarp, it is called Cleistothecium. In exams question we can expect like this. So completely closed ascocarp is called as Cleistothecium, Apothecium, Perithecium, Gymnothecium, or naked, something like that they will ask. Okay, if it is a completely closed ascocarp, where see there is no opening here, where somehow when the maturity arrives, automatically it will burst and it will release outside. That's secondary. So but when you see here, it is completely closed. So completely closed ascocarp is produced by Cleistothecium. Oh, for example, Cleistothecium is produced by the fungi like powdery mildew, penicillium, and all those things. I mean, all the fungi can be actually produced from Cleistothecium. When it comes to Apothecium, it is a partial, I mean, completely opened ascocarp. Example, Helvella, Pezizales, all this fungi comes under. I mean, can be produced from this Apothecium. When it comes to Perithecium, that is partially opened ascocarp. So this is Perithecium. We can inside we can see some spores, right? So inside the sac spores are there. Actually, spores like Xylaria, Nectaria, or maybe Hypocreals, Claviceps, all are produced from this Perithecium. When it comes to naked SI, it is like without fruiting body. There is no fruiting body here. That's why it's called as naked SI. For example, Taphina can be produced by produce this type of thing. And not fully or partially closed ascocarp is Gymnothecium. As I told you, that Thalleromyces or Arthroderma can actually produced by produced from this Gymnothecium. So the next one is different types of hemathecium. Why? What is hemathecium means? Internal ascus tissues. As I told you, this is a flask-shaped structure. Internal ascus tissues were arranged in different arrangements. These different arrangements, internal ascus tissue. Now we are going to discuss about hemathecium, otherwise called as internal ascus tissues. Okay. Here 5 types are there: paraphysis, paraphysoids, periphysis, and pseudoparaphysis. I will tell you in a simple way. Paraphysis means hyphoarginate. See here, this is the one. Paraphysis means hyphoarginate from the base. This is the base, right? This is the base. Actually, hyphoarginate from the base and grow between asci. So here actually hyphoarginate from base. So actually develop in between asci. For example, Xylaria. So paraphysoids, this is paraphysoids. It is resembles like a pseudoparaphysis with septa. It resembles like pseudoparaphysis with septa. Example is Patellaria species. The next one is periphysoids. Periphysoids means you can see finger-like projections which actually develop in downward direction, not upward direction. Actually, you can see here these finger-like projections actually developing downward direction, not upward direction. You can see here these finger-like projections are developing actually to downward direction from the ostiole. Example is Nectaria. Periphysis hyphae actually develop inside the ostiole. You can see here hyphae develop inside the ostiole and come outside. It came outside, so ostiole opening, so it will come from ostiole to ostiole opening. This is called periphysis. Example Zibrella. And pseudoparaphysis means hyphae originate above the asci here. Hyphae originate above the asci and grow downwards between the developing asci Example is Pleospora. These are all the different kinds of hemathecium. Hemathecium means internal ascus tissues only. We can expect by giving the definitions. So finger like projections which actually develop from the downward direction is an example. Which type of hemathecium? Periphysites, paraphytes, something like that. There might be a chance of asking. You just go through. So the next one is we know very well each ascus produce 8 number of ascospore. Generally, you can, if you see ascospore or an ascomycota group, all ascomycota fungi produce 8 number of ascospores. Now there is some changes or some variations. Now we will see. So normally few fungi produce only 2 ascospores. So 2 ascospores is produced by Phylactinia. 4 ascospores is produced by Neurospora tetrasperma. Neurospora tetrasperma. Next one is 5 ascospores are produced by Capnodium. 8 ascospores are generally, almost all ascomycota group of fungi produce 8. We know very well, right? And 16 ascospores are produced by Trichoderma, that is otherwise called as Hypocrea rufa. This Trichoderma actually produce 16 ascospores, okay? And 512 ascospores are in a single ascus, that is produced by Podospora. So far we have seen ascospores. Now I am telling one ascus, only it produces one ascus in a fungi. So one ascus producing fungus is Podosparia. Podospora is different, Podosparia is different. Podosparia leucotricha and Sparotheca panosa, rose powdery mildew. So this Sparotheca panosa and Podospora leucotricha can produce the only one ascus. Different types of powdery mildews. So powdery mildews of 3 types, that is oidium, oidiopsis, ovuleropsis. These are different types of powdery mildews we can see. So I have given some differences, so I will just tell you the important points only. So the remaining things, if you want to refer, you just go through, otherwise you can just ping me in the comment box. So leave out all the symptoms and all those things. So let me tell you here, mycelium in oidium type, mycelium is septate and ectopytic. We can see here in mycelium septate, here we can see mycelium is a septate and ectopytic. And when it comes to oidiopsis type, mycelium is septate and endopytic, where mycelium is endopytic. We can see here mycelium is endopytic. When it comes to ovuleropsis type, septation is ecto and endopytic, both ecto and endopytic. When it comes to conidiopore is a club shaped non-septate. Here club shaped non-septate. Conidiopore is club shaped non-septate, whereas oidiopsis type of conidiopore is long branched and septate. We can see here septations, right? And when it comes to branched and septate, here it is branched and septations can be seen. So Cleistothecia, the appendages of Cleistothecia is actually powdery mildew. All these things can produce it from Cleistothecia, that was discussed in sexual fruiting bodies. So this Cleistothecia produces mycelioid appendages, whereas oidiopsis also produce mycelioid appendages, but ovuleropsis type produce bulbous appendages. The appendages are bulbous in ovuleropsis, and mycelioid in both oidium and oidiopsis. The most important thing is example. So oidiopsis type is produced by Ericifae. Oidium type, not oidiopsis. Oidium type is produced by Ericifae. Idiopsis type is produced by Levilulla or Levilulla, and ovular abscess produced by Phylloxera. They can ask examples like ovular abscess. Give an example of host that is ovular abscess type of powdery mildew is produced by Phylloxera. They can also ask questions like this: idiom type of powdery mildew was produced by, that is Levilulla. The examples are very important, and other description has been given. You guys just please go through. So the next one is type of appendages in powdery mildew. So now we will discuss about the type of appendages of powdery mildew, that is various type of appendages on the Cleistothecia. So first of all, let me tell you what is Cleistothecia. Cleistothecia means it is a completely closed ascocarp where the asci are produced inside. Whenever the time come or maturity at the stage of maturity, a pressure will be produced from the inside and it bursts out, and the spores are released outside. So here Cleistothecia we can differentiate into one ascus type or many ascus type. Okay, one ascus means it contains only one ascus, many ascus means it produces many ascus. So in one ascus, mycelioid appendages or mycelioid type is produced by Sparotheca. We can see here mycelioid. So here it is a mycelioid. Sparotheca, it produces only one ascus. Appendages are mycelioid appendages. You can see the appendages are mycelioid appendages. So the next one is the dichotomously branched. Dichotomously branched is produced by one ascus, with the dichotomously branched is produced by Podospora. You can see here the appendages are dichotomously branched. If you see, all the branches are dichotomously branched. Same. See, one ascus with mycelioid means Sparotheca. One ascus dichotomously branched means Spodospora. The next one is the circinide or coiled tips can be observed in many ascus. You can see many ascus. So many ascus are there. These are all the tips are coiled. You can see here all the tips are coiled. Circinoid or coiled tips are can be seen in Uncinilla. I have seen this question 2 times in net. Circinoid appendages or coiled appendages is the characteristic feature of Uncinilla, Phylloxera, Erysipeia, something like that they will ask. Okay. So here Uncinilla produces circinoid or coiled type of appendages. So the next one is a bulbous appendage. You can see here at the starting of the appendages, it look like bulb-like appendages, onion bulb, right? Example I am telling. It look like bulbous appendages. So many ascus with bulbous appendages are produced by Phylloxera. That is Phylloxera. Okay. And the next one is mycelioid appendages. Many ascus with mycelioid. Single ascus mycelioid means Sparotheca. Sparotheca. Many ascus with mycelioid is produced by Erysipeia and Levilulla. You can see many ascus with mycelioid. It is also like mycelioid. So many ascus with mycelioid produced by Erysipeia and Levilulla. Dichotomously branched. Many ascus with dichotomously branched. So single ascus with dichotomously branched means Podospora. So many ascus with dichotomously branched is Microspora. So we can definitely expect a few questions something like this. So the next one is type of smuts in sorghum. So I can't able to explain all this table, that I will tell you probability questions or what kind of probability questions we can expect. So the maybe pathogens they will ask. Grain smut is caused by, or head smut is caused by, or loose smut is caused by, or long smut is caused by. So grain smut is caused by Sphaerotheca swargi, head smut is caused by Sphaerotheca riliana, loose smut is caused by Sphaerotheca cruenta, whereas long smut is caused by Toliposporium ehrenbergi. Here I think names are changed now. Now new names came, I think. The next one is what we can I expect is ear infection. So ear infection, all the grains are infected or some grains, something like that. Most of the grains are smutted in grain smut. So when it comes to head smut, entire inflorescence is converted into a big sporus. Or when it comes to loose smut, all or most of the grains are smutted. When it comes to long smut, very few grains are infected. The questions will be like this: in which smut inflorescence can be infected? So if you see other smut, inflorescence cannot be infected, but head smut infected sorghum, inflorescence can be infected. I think I have seen this question, but not in a net exam. but somewhere in the entrance exam I have seen, maybe very long time back, I think so. So inflorescence can be converted into smut spores. What is the characteristic feature of head smut, or maybe this kind of questions we can expect. Site of infection of head smut is head smut ovary can be infected, as I told you. So head smut inflorescence, grain smut ovary, head smut inflorescence. Again loose smut and long smut is ovary. So except head smut, all of site of infection is ovary, but only head smut, the site of infection is inflorescence. And maybe they can ask columella is present or absent, something like that. So in grain smut columella is present, whereas head smut columella is absent. Again loose smut columella is present, again long smut columella is absent. Most important thing I have seen is spore viability. So grain smut spores can survive more than 10 years in the soil. So they can ask questions like which of the following smut can survive more than 10 years in the soil, something like that. They can ask. Okay, I have some small technique for you to remember this. I will tell you what about that. So for grain smut spore viability is 10 years, and head smut spore viability is 2 years, long smut spore viability is 4 years. Again sorry, loose smut spore viability, long smut spore viability is 2 years. How can we remember? You might have a doubt, how can we remember this years by like. Before that, you guys need to remember, let me tell you, give me an example. Imagine it is a sorghum. Okay, starts with the grains. So grains are there up on the I mean in head grains are there, right? So first with the grains, you can, this order is very very important. Grain smut followed by head. So starts with head, right? Head, then loose smut, then long smut. So you can maintain this order: grain smut, head smut, loose smut and long smut. Let me tell you, our education starts with first class to 10th class, 1 to 10. So 10 years we are studying from 1 to 10, right? Again 2 years plus 1 and plus 2, and again 4 years for BS agriculture. We studied BS agriculture, right? 4 years. And again this 2 years is for MSc masters. So after MSc we need to for graduation we will go for MSc, right? So this you just match this with this order: grain smut, head smut, loose smut and long smut. So grain smut 10 years, head smut 2 years, loose smut 4 years and long smut is 2 years. Okay, this is the order. So for all smut can be managed by seed treatment Vitavax. So in general, there are something about this Aspergillus penicillium and will be there. I will tell you. So generally, black mold. You can see generally black mold in Aspergillus is Aspergillus niger. Green mold. Generally, green mold in Aspergillus is Aspergillus flavus. White mold is Aspergillus candida. Brown mold is Aspergillus tamari. Yellow mold is Aspergillus aureus. So questions I have already seen the questions from this section. That's why I just placed black mold of Aspergillus, or black mold is Aspergillus niger, green mold is Aspergillus flavus, white mold is Aspergillus candida, brown mold is Aspergillus tamari, whereas yellow mold is Aspergillus aureus. When it comes to the penicillium, blue cheese fungus, that is Penicillium roquefort. Actually blue cheese is actually produced from this Penicillium roquefort. Okay, blue mold of penicillium. It's not blue mold of Aspergillus. It's a blue mold of penicillium. It is called Penicillium italium. Green mold of penicillium. In Aspergillus also we have seen green mold. When it comes to Aspergillus green mold, that is Aspergillus flavus. When it comes to Penicillium green mold, it is Penicillium digitatum. Okay, the rot of apples is caused by Penicillium expansum. In general, it is not related to Penicillium or Aspergillus. In general, if it is a grey mold fungus, that is Botrytis. Mostly we can see on strawberries. You just buy strawberries, place it outside or maybe in the fridge. Within 2 days, you can see the Botrytis. Very severe pathogen in especially this strawberries. So black mold in general, Rhizophus stolonifer. White mold fungus, Sclerotinia sclerotiarum. Peat mold fungus, that is Pezziza ostracoderma. This is in general. We may expect maybe probability we may expect. Now we will see something about Fusarium. So which normally causes wilts. So here Fusarium wilt of banana is otherwise called as Panama wilt. or banana wilt. So which is caused by Fusarium oxysporum pharma species cubensis actually survive in the soil. So this Panama wilt is a complex disease which is caused by Fusarium oxysporum pharma species cubensis, which is actually also associated with one more nematode called banana burrowing nematode, that is Rhadophilus similis. So here two questions we can see. Banana Fusarium wilt of banana is caused by Fusarium oxysporum pharma species cubensis, or which nematode is associated with this banana wilt, the Rhadophilus similis. So with the help or association of this nematode, the severity of the banana wilt is actually goes up, increases, okay, and this can be effectively managed by the application of lime at the rate of 2500 kilograms per soil. This is one of the management practice. And the next one is Fusarium race T affects grass Mitchell or grass Michael. Fusarium race 2 affect bulgo, cooking, that's a cooking banana variety. And Fusarium race 3 affects Heliconia, and Fusarium race 4 affects Cavendish. In the meantime, in bacteria also they have a Raustolinus solanacearum race 1, race 2, race 3, race 4, race 5. You guys will see in bacteriology section, okay. So here last year this question was asked, 2021. If you want, you just go and see in the section of our already published videos, and they asked the question like this: Fusarium race 1 which completely wiped out or which completely affect which of the following variety, something like that they have asked. Grass Mitchell or grass Michael, whatever you want to pronunciate. Here the most important thing is, we know very well that Fusarium can produce three kinds of spore: microconidia, macroconidia and chlamydospores. But here Fusarium oxysporum pharma species cubensis, that is TR3 race 3, cannot produce or do not produce any chlamydospores. It don't produce any chlamydospores, okay, and it is actually a diversified pathogen, we will see. So wilt of tomato is caused by Fusarium oxysporum pharma species Lycospores. See, wilt of cotton is caused by Fusarium oxysporum pharma species Vasinfectum, and wilt of banana is caused by Fusarium oxysporum pharma species Cubens, okay. So it's a very important one. Let's discuss about wheat stem rust. So before entering into the in detail about wheat stem rust, let's have some technical terminologies here. What is microcyclic, macrocyclic, demicyclic, autocyclic? So microcyclic means all the 5 stages are absent. Actually, if you see rust, 5 stages are present: pycnospores, ascospores, iridospores, teleospores, basidiospores, something like that. 5 spores will be present. Micro means all 5 spore stages are not present, or maybe missing or absence of any one of the stage, maybe more stages also, okay. And macrocyclic means all the 5 stages of spores are present in the life cycle, like pycnospore, ascospore, iridospore, teleospore, basidospore, as I told you na. So all the 5 spores are present in the macrocyclic. Demicyclic means lack of iridial stage or iridospore is called demicyclic. Last year also this question was asked. Very important. Out of 4 net exams that I wrote, I think this question was there almost 2 times. I think, I'm not sure, maybe 2 to 3 times. If you see the question paper since 2009, this question was asked repeatedly many times. Lack of iridial stage is called demicyclic rust. Demicyclic means on the lower side, like they will give options like lack of iridial stage, lack of facial stage, lack of pycnial stage, lack of teleospore, something like that they will ask, okay. Demicyclic means lack of iridial stage. Very important. Autoecious means the pathogen completes entire life cycle in a single host. Autoecious means the pathogen complete life cycle in a single host is called autoecious. When it comes to heteroecious, the pathogen requires genetically 2 different hosts to complete the life cycle. For example, if we take wheat stem rust, so the main host is wheat, alternate host is barberry, okay. Why I am calling it as alternate host? Because alternate host is a host species which actually belonging to the different species or different family of a main host. Because if you see, wheat comes under triticales, whereas barberry comes under some other family. That's why it's called as alternate host, which is belonging to different family, okay, not same family. So when we see here autoecious example of, we can also expect this example of autoecious macrocyclic rust. Example of autoecious macrocyclic rust: bean rust, flax rust, sunflower rust, safflower rust, castor rust. All rusts are the example of autoecious macrocyclic rust. That's why I give BFSS. B stands for bean rust. F stands for flax rust, S stands for sunflower rust, again S stands for safflower rust, C stands for castor rust. So these all rusts are example of autoecious macrocyclic rust. Now we will see what is autoecious. The pathogen complete the entire life cycle in a single host. What is macrocyclic? All 5 spore stages are present. So all 5 spore stages will be finished in a single host. That is called autoecious macrocyclic. Autoecious demicyclic. Demicyclic means lack of radial stage. The pathogen complete life cycle in a single host, but the iridial stage is absent. Demicyclic means lack of iridial stage, right? Example is rubus orange rust, which is caused by Gymnoconia peckiana or Gymnoconia nitens. Heteroecious macrocyclic rust, very very important issue. Heteroecious means the pathogen requires genetically 2 different hosts to complete the life cycle. Macrocyclic means all 5 spore stages of a life cycle of a all 5 spore stages of a pathogen is present. Heteroecious demicyclic macrocyclic rust, example is wheat stem rust and white pine blister rust, that is caused by Cronartium rubicola. It's a very very important. The next one is heteroecious demicyclic rust. Heteroecious means the pathogen requires genetically 2 different hosts to complete the life cycle. Demicyclic means lack of radial stage. Okay, so heteroecious demicyclic rust example is cedar apple rust. We also called as car. You can remember the driving car, something like that. Okay, car means cedar apple rust, which is caused by Gymnosporangium juniperi verizinane. So alternate host for cedar apple rust is apple. As I told you, demicyclic means lack of iridial stage. Lack of iridial stage is called demicyclic rust. Very important. So these are some basic terminologies. So we can't able to discuss life cycle, but I will tell you some important stages in the life cycle of stem rust. Okay, we will see here. As I told you, 5 spore stages are present: pycniospore, asciospore, iridospore, teleospore and basidiospore. So pycniospore can be denominated as 0. So actually in the life cycle of wheat stem rust, asciospore, iridospore, teleospore and basidiospores are initially discovered, and actually the pycniospore were discovered much more later. Okay, here the pycnial stage or pycniospore were first discovered by Craig, Craigie, Craig in 1927. So Craig is the scientist or the man who first discovered the sexual stage, that is pycniospore. That's why it's named as 0, because it is a very crucial connection to continue the life cycle. So this pycniospore denominated as 0. Okay, it produces number of germ pores, germination pore. How many number of germ pores it can be? So pycniospore will produce 1 germ pore, asciospore produce 6, iridospore produce 4, teleospore produce 2, whereas basidiospore produce 1. So if you see, pycniospores and asciospores are actually hyaline. So pycniospores are hyaline in color, asciospores are yellow in color, iridospores are golden brown in color, teleospores are dark brown to black in color, or basidiospores are hyaline in color. So pycniospores are haploid, asciospores are dikaryotic N+N, iridospores are also dikaryotic N+N, teleospores are dikaryotic and diploid N+N and 2N. Okay, basidiospores are haploid. So if you see this pycniospores and asciospore, these spores are actually happened in alternate host, that is Barbary. So imagine, so Barbary is the alternate host for wheat stem rust, right? So this pycniospores and asciospore can develop on the secondary host, that is alternate host Barbary only. These spores cannot germinate on a main host, that is wheat. So pycniospores and asciospores can germinate on Barbary only. Okay, Barbary is alternate host. So pycniospores actually produced on the upper surface. If you see this in sexual reproduction also, we have seen spermatia, right? Spermatia, from spermatia where pycniospores are developed, and exactly to the corresponding lower surface asciospores are developed. Okay, that's why pycniospores are upper surface, asciospores are lower surface. So both are happens in an alternate host, that is Barbary, right? So iridospores and teleospores are generally produced in wheat only. That's why upper surface only. If you see the leaf surface of leaf of wheat, iridospores and pycniospores are produced on the upper surface only, that is primary host. So basidiospores can happen only in soil debris. It won't germinate on anywhere. It survives in only soil debris. Okay. So this is something about rust. Let me give some other information. So rust generally produce iridospore. Iridospore is otherwise called as a repeating spore. Iridospore is otherwise called as repeating spore, otherwise called as summer spore, which is actually n+n in condition, where this iridospore are golden brown in color, which is actually iridospores are single cell. For the management of rust, we can recommend carboxin. The trade name of carboxin is plant wax. So this is the order, that's why I placed it. Again I coming, rust produce iridospore. Iridospores are repeating spores which continuously produces, right, continuous reproduction will happen, that's why it's called repeating spore. Okay, and it is a summer spore, n+n condition, that is dikaryotic, and the color of iridospore is golden brown, where it is a single celled, and the management is oxycarboxin. The trade name of oxycarboxin is plant wax. When it comes to smut produce teleospore. Teleospore is actually a resting spore, and the one more thing about teleospore is the bicell, that I will tell you later. Okay, this otherwise called as winter spore, and the condition is n+n or diploid also. And actually the smut spore, that is teleospore, is black in color or sometimes dark brown in color also. It is actually bicelled. In the entire life cycle of wheat stem, the only one bicelled spore is teleospore, direct bicelled spore I am telling. Okay, dikaryote, not dikaryote, bicelled spore. Okay, and it is otherwise called as teltospore also. For the management of sort, we can recommend carboxin. The trade name of carboxin is Vitavax. Rust localized infection or foliar parts can be seen, whereas smut systemic infection that can mainly seen on inflorescence as well as grains. Okay, so the types of teleospore, this is very important, that you can take last 10 years net question papers except 2021, there must be a one question from this. Okay, so we will see the types of teleospore. Single celled teleospore is produced by Euromyces, Melanospora, Pacospora. See, Euromyces, see this actually Melanospora and Pacospora are generally the type of teleospore is actually single celled only. Okay, this Melanospora and Pacospora also comes under some other type of teleospore, I mean some other kinds, but here I have shown the crust like teleospore Melanospora, right? Even though it is a crust like, but it is single celled only. Okay, that's why I placed it here. So single cell teleospore is produced by mainly Euromyces, but Melanospora and Pacospora is also a single celled type of teleospore only. Okay, double celled, as I told you, double celled teleospore is produced by Puccinia, very important. So single celled, how it will be look like. So double celled teleospore, that is double celled. Okay, and multi-celled, number of cells, multi-celled teleospore is produced by Phragmidium. This is a multi-celled teleospore. When it comes to turnip shaped or napiform shaped teleospore or kidney shaped iridospore, here Emilia, right? See, kidney shaped iridospore, turnip shaped teleospore, both are produced on the same fungi. Okay, so turnip shaped teleospore or napiform shaped teleospore or kidney shaped teleospore, otherwise called as hump beaked teleospore, which is produced by Hemilia, which causes coffee leaf rust, Hemilia vastrotrix. Okay, crust like teleospore, you can see here, this is like a crust like teleospore is produced by Melamsora. Sorry, this is a crust like teleospore is produced by Melamsora. This is Melamsora. The last one is parachute like teleospore is produced by Ravinelia. This is Ravinelia. So the spore is actually looking like a parachute, size is the parachute, that's why it's called as Ravinelia. So okay, we can definitely expect one question from this. So the next one is alternate hosts and collateral hosts of some pathogens has been given. I am telling you what is alternate host means. Alternate host is the secondary host where the main host and the alternate host is not belonging to the same family. If it is a different family, it is called alternate host. Main host and secondary host is different family, that's why it's called alternate host. If the host, main host and the secondary host is of same family, it is called collateral host. Collateral host is otherwise called as alternative host. There is a difference between alternate and alternative. Alternate host means it is of same family. Alternative host means different family. Alternative host is otherwise called as collateral host. Let me tell you. Wheat stem rust, Puccinia gramineous etc. Primary host is wheat, secondary host is barberry. When it comes to leaf rust or ground rust, Puccinia recondita, primary host is wheat, and it is believed to be Thalictrum species is the secondary host or alternate host. Sorghum rust, Puccinia purpurea, sorghum is the primary host. Sleeping beauty, that is Oxalis corniculata, is the secondary host, that is alternate host. Pale millet rust, Puccinia substriata, pale millet is the primary host, brinjal is the alternate host. Maize leaf rust, Puccinia sorgi, maize is the primary host, Oxalis is the secondary host. When it comes to pine blister blight, Cronatium rubicola, currant is the primary host, pine is the secondary host or alternate host. Cedar apple rust, Gymnosporium junipero verizinare, apple is the primary host, and again I'm sorry, maybe it is little bit wrong, I'm not sure, but apple is the primary host that I know. Maybe alternate host of apple, maybe actually I have no idea. Please let me come to know. If you guys know, just please comment below. And the crown rust of oats, which is called Puccinia coronata, sorry. Primary host is oats, secondary host is buckthorn, that is Rama species, and others. So when it comes to, now we are discussing about collateral host or alternative host. Alternative host means which is of same family. Main host and secondary host is belong to the same family. Here rice and Lersia exandra are of grass species. Lersia exandra actually grass species which are both are belonging to the same family, that is Gramineae or Poaceae. Okay. Rice blast, Pyricularia varizae or Magnoporthia graceae. Primary host is rice, whereas Lersia exandra is the alternate host, where this rice and Lersia exandra are belong to the same family, that is Poaceae or Graminae. Bacterial leaf blight of Xanthomonas varizae, Pathogen varizae. Primary host is rice, whereas Lersia exandra, Cyprus rotundus can act as an alternative host. Downy mildew of maize and sugarcane, which is caused by Pernosclerospora philippiensis or Pernosclerospora saccari. Maize and sugarcane is the primary host, and where alternative hosts are sorghum halapen, sorghum bicolor, and saccharum spontaneum is the alternative host. Downy mildew or green ear of kombu, which is caused by Sclerospora graminicola. The primary host is bajra, kombu or pal millet. The secondary host, that is alternative host, is echinocola and panicum species. Coming to the last one, sugarcane whip smut, which is caused by Sporisorium setaminae. Before it is called as Stulagnosetaminae, now it is called as Sporisorium setaminae. And the primary host is sugarcane, and the alternative host is Saccharum spontaneum. So coming to the different forms of rust based on the absence or presence of spore stages, we may also expect. So zero stage means pycnium or pycnelium, pycnial stage. One means acium or acial stage. Two is iridial stage. T-teleal, four basidial. We know very well. So if it is a U type means 0, 1, 2, 3 stages are present. Fourth stage is absent. That is basidial. Brachytype means 0, 1, 2, 3 stages are present. 0, 2, 3 present, but 1 is absent. 1 means Asian. So Opisis type means 0, 1, 3 is present, but 2 is absent. 2 means iridial stage. Hemi type means 2 and 3 are present, 0 and 1 are absent. 0 and 1 means pycnium and Asian. Okay. Microtype means only 3 are present. Leptotype is also only 3 are present. 3 means only, that is a teleal stage is present. Okay. So, and we discussed earlier that Ascospore contains 8 number of ascospore, generally all ascomycota. But when it comes to basidiomycota generally contains 4 number of basidiospores, but few other basidiospores exceptional that can produce more number of basidiospores, more than 4. Let me discuss. So 9 basidiospores are produced by Pallas impudicus. 2 to 8 number of bastiospores is produced by Exobasidium vexans. 8 to 16 number of bastiospores is produced by Tilicia caris. 14 to 30 bastiospores are produced by Tilicia controversa, and 32 to 128 bastiospores are produced by Tilicia indica. These are all comes under Basidiomycota only. Okay. So the next one is the kernel bunt of wheat, which is caused by Tilicia indica, before previously called as Neovacia indica. So this genus Neovacia was named by I think B.B. Munkur. Yes, here is there. Yes, Neovacia, the genus name was given by B.B. Munkur. So the kernel bunt of wheat is called by Tilicia indica. It was first reported by Mitra in 1931. In kernel, the area of first identification is kernel, that's why it's called as kernel bunt. Okay, so the kernel bunt infected fields produce rotten fish. It's very, very important. I have seen this question many areas, many places. So the rotten fish, because the infected fields produce trimethylamine. Due to the presence of trimethylamine, it produces rotten fish smell or alcohol smell. Okay, this kernel bunt is otherwise called as partial bunt, because only few ears or few grains are infected. That is partial infected, not completely. That's why it's called as partial bunt also. Okay.
So the spore types are conidia types, that is secadian classification. If it is a single celled aseptate, it is amroconidia. Two celled means didymoconidia. Multi-celled spore means phragmoconidia. Star shaped conidia is staroconidia. Multi muriform septation can be seen in dictyospore. Both vertical and horizontal septations. So again one more example, both vertical and horizontal septations are muriform conidia can be observed in alternaria also. Alternaria conidia also produce muriform septation, that is both vertical and alternate septations, horizontal septations. Piliform or wormy shaped is scolicospores. Helical spore or spirally coiled spore is helicospore. That is scolicospore, sorry. That is scolicospore, that is helicospore. And a hand-shaped spore with several more or less parallel or finger-like can be thin cherospore. So this is also important. I think this one question was asked from this lab for 2021 net exam. I think so. I think so. So the next one is mode of entry of fungal pathogen. Here I differentiated for simple understanding. So mode of entry in bacteria, I have clearly given, clearly explained in detail about this mode of entry of plant pathogens. So let's discuss. So mode of entry of fungal pathogens. Stomata are natural opening. So Puccinia graminis triticea and Pernospora destructor, that is downy mildew and powdery mildew, actually enters into the plant cells through stomata. Wounds, the pathogens, what are the pathogens that can enter into the through wounds? That is Rhizopus, Gliosporium, Aspergillus, Penicillium, Coltrotrichum, Diplodi, etc. And the lenticels, Sclerotinia fructicola, Phytophthora aricae, powdery mildew, and potato scab, apple fruit, all these are enters into the plant cells through lenticels. Cuticle, rust, downy mildew, powdery mildew, and botrytis can enter into the plant cells through cuticle. Seedlings, grain smut of jawar, loose smut of jawar, like wheat bunt disease. So these are all kernel bunt of it all enters into the plant cells through seedlings. Root hairs are scaly hairs, wilt, that is fusarium, club root of cabbage, root rot of cotton, sugarcane, whip smut, all enters into the plant cells through root hair or scaly leaves. Buds, peanut rust, flowers, loose smut of wheat, and like a bunt of rice, ergot of rye, long smut of jawar, all are enters into the plant cells through flowers. And leaves, white pine blister blast, nodes, sugarcane red rot, and epidermal cells Phytophthora infestans, coleoptile wheat stinking smut or bunt, and seed coat pythium, stigma Aeschylocosetamina, that is the smut of sugarcane, claviceps fusiformis and Disclocon rotatritici, and insects, Dachelum disease, that is Cetrocystis ulmi. Stalk ends, Penicillium italicum, Thallus paradox. These are all the various fungal pathogens and their mode of entries. When it comes to bacteria, Hydrothos black rot of cabbage and bacterial blade of rice, and lenticels potato scab, trichomes bacterial canker of tomatoes, tomato bacterial leaf stick, citrus canker, and angular leaf spot of cotton, and insects bacterial wilt of corn and bacterial wilt of cucumber are the very important. Where bacterial wilt of corn is transmitted by corn flea beetle, that is Chitrinum policaria, whereas bacterial wilt of cucumber is transmitted by cucumber beetles, that is Diabrotica victata. Now it is called as Achylema victata. Another species is Diabrotica duodecim punctata. So coming to the sex hormones, as I told you that fungi can reproduce by means of sexual and asexual reproduction. So here a few of the hormones that is produced by fungi, that is sex hormones. Now I will tell you these are all the various kinds of sex hormones, that is cyrenin, anthradiol, oogoniol, trisporic acid, yeast alpha factor, and perisinins. Let me tell you, this cyrenin is the first discovered sex hormone. This is the first discovered fungal sex hormone, cyrenin, which is produced by Allomyces. And anthraδιol is produced by Achilles species. Oogoniol is produced by Achilles species, that is Achila heterosexualis. And trisporic acid is produced by the members of Mucorales, that is Mucoromycido and Blackslea trispora. And yeast alpha factor is produced by Saccharomyces, that is Saccharomyces cerevisiae. These are all the various species and their specific function has been given here. So mostly we can expect the cyrenin is produced by, or what is the first plant, what is the first fungal sex hormone to be discovered, that is cyrenin, and produces species also, that sometimes they may ask. So these are all the important concept and all those things about mycology. So hope this will be useful, and if there is any doubts and all those things, you guys can comment on the comment section. Stay geeky, stay tuned. We are Team Geeky Researchers.